Hello, everyone. Here we are again. My name's Ian Deary. I'm director of the Lothian Birth-cohorts with my co-directors Joanna Wardlow and John Starr, and the study coordinator at the end there, Dr. Simon Cox. So welcome back. After, I think, 4 years we met here last. It's nice to see you again to celebrate a reunion of the Lothian Birth-cohorts of 1921 and 1936. Now, we've got a lot on for you today, so let me give you the rundown of what's going to be happening in the next few hours. So first of all, by the way, there is no fire drill planned. So if the fire alarm goes, get out, okay? That's it. Right, so I've done something unusual today. I decided to mix it up and pit the 1921s against the 1936s. Yeah. And you can't tell who is who, can you? So 21s, where are you? Great. And the rest of them are 36s. Okay, right. Now, what we're celebrating today is, as you know, the end of what we call the field testing. It takes quite a while to get through everybody, and at the end of that, I like to bring you in and show you the results before anybody else gets to see them, including fellow scientists. So I'm going to be showing you some of your results and how you've progressed from 70 to 73 to 76, and from, if I can remember this, the 21s from 79 to 83 to 87 to 90 to 92. So that's what we're doing today. So you'll get first view of them. I'll speak first until about just after past, and then Professor Wardlaw will talk about the brain imaging in the 1936s, and John Starr will talk about the medical results in the 1936s, and then you'll be seeing a short film that a professional filmmaker has made about the study. Then we'll have a break for tea, and we want to replicate do you remember the photograph we took on the steps outside? We want to replicate that, so be careful, no rushes, but it would be wonderful to do that. Oh, by the way, people often ask me, how representative is this sample of the population? And I say, well, we're only interested in people who can make it up 37 steps at New College. I did go and count them, actually. I was hoping it would be 39, then I could have made a proper joke, but anyway, never mind. So you're going to have some tea, and we'll do the picture at Simon at the beginning of the tea break. Then you'll get tea, be able to have a blether with the team. And I've asked Simon to put together some posters. So members of the team will stand beside posters about what they do. So go and blether to them. They're there to speak to you, not to each other. I'll be watching for that too. Then after the break, we're going to be doing 4 5-minute talks of some new things that we're going to be doing in the study that I hope you'll find exciting, and then we'll have a long time for questions. Now, during the question time, do you know that I usually ask you to try and write down some difficult questions that Simon, that used to be Alan, will throw at me, and that myself and John Starr will have to answer after the break. So do that. Simon, where's the paper and pencil for that? You'll tell him at the break?
I will do just in that corridor there.
Just outside. So we'd like as many of you as possible. We've got lots of pens, lots of pencils, bits of paper. But also, Simon's going to ask you to do a second thing as well. Do some of you know, you should know, that there's this British Academy debate happening in a couple of weeks' time. Who's planning to go to it? Oh, brilliant. Well, I'm speaking at it, so I better put my hand up as well. That is about the best years of our lives. And one of the things I'd like you to do is, on the back of the paper, if you do ask a difficult question or an easy one, is actually tell us when were or when are the best years of your lives, and why as well. Okay, that could be now, it could be sometime before that, or sometime to come. But I better get on with what I've got to say today. So, welcome to those in the Lothian Birth-cohort 1921, and welcome to the youngsters in the Lothian Birth-cohort 1936. And this is to remind me that in question time, please do take the opportunity not just to ask us questions, but to make suggestions about things you would like to see us do in the study. And that's actually worked before, because you have suggested things during the question time that we've actually taken on board, and we've changed the study as a result. typically added more in for you to do, but you can blame your colleagues for that. Right. And one of my young researchers said, you think the LBC are famous, but there's actually another LBC that's better known than you. And I'd never heard of them, I have to say, but they have their own logo. And there they are, the LBC crew. And I don't actually see them in the audience today, this particular LBC, but you've come along and we've been here before. Well, actually, we've been somewhere else called the Assembly Rooms before. So if anybody is sitting in the Assembly Rooms today, they're in the wrong place because we're in the Assembly Hall. Who remembers this? Good. That was the original gathering of the Lothian Birth Cohort 1921. Was that in 2000, 2001? When we first started, we started it in 1999, and that's the hardest thing we ever did, was try and serve 550 afternoon teas to a group of individuals taking part there. So you won't be getting a sit-down afternoon tea with scones today. But after that, you'll recognise, do you remember our first meeting? The Lothian Birth Cohort 1936 here. And then we met again. And then there you are on the steps. And then there was the last one. And just to remind you, there's some of the team that tests you at the Wellcome Trust Clinical Research Facility. And who can spot the only constant in each of those photographs? Yep, Janie, okay, who's in all four of them. Right, why are we here? Well, why we're here is you are extraordinarily unusual. Having taken, as part of the Scottish Mental Survey in 1932 and in 1947, you had these well-validated mental test scores from age 11 that we are using as the baseline for studying cognitive change into older age. Oh yeah, that's my first controversial slide. And do you remember, I showed this before to the 1936s, but you slightly outdid the 1932 mental survey when you took the test in 1947. So for some reason that we still don't understand, the people born in 1936 had an average of 2 points better on that mental test that you took at age 11. Whether you were better fed or paying more attention on the day, we still don't know. Maybe you had practiced on the test. I do hope not. And then we discovered these data in St. John Street at Murray House in the late 1990s. And there they were, the ledgers that we still use today from the 1932 mental survey and the 1947 mental survey. And that's what those of you who were born in 1921, the pages of the books looked like. Of those of you born in 1936, that's what the pages where your data are stored look like. I've covered up the names, obviously. Now, the aims, just to remind you, what we're interested in is why do some people have better aging of thinking skills and brain health than others? And people range from those who've got very successful mental aging to those who have slightly more mental decline. And the thing that's very unusual about you and that the world now knows about is that you have this information from age 11 that gives us the baseline in youth from which to study the changes into old age. Now, some information about the numbers. For those of you in 1936, we first saw you at age 70. That is the problem. I do know what age you all are, so I'm afraid that there's no demure hiding of ages in here except about amongst us staff. Oh, by the way, yeah, this got snuck in. Do you remember this as well? The team sprung me on this when I first saw you in the 1936s. When we saw you at age 70, we hadn't by then scanned your brains, and Professor Wardlaw was then brought in as a co-investigator to lead the brain imaging side. And so when we saw you at 73, brain scans started. And then we've completed the age 76 with mental testing, physical testing, and brain scanning once again. And there are the numbers. So this is the first of the new information that we're showing you for the first time and showing it to you as the first people to see this. So there are 1,091 of you at age 70 in the 1936, 866 at age 73, and 697 at age 76 on wave 3, it should say there. Now, in terms of the brain scan numbers, there are 1,091 came to wave 1, and of those at wave 2, 728 were brain scanned at wave 2, and 488, almost 500, at age 76. And do you remember the test that you got in the neck, where they scanned, Professor Wardlow's staff scanned the neck? That was 821. Now, do you remember the brain tissue bank that we asked you about as well? Well, actually, I carry this telephone, and if that goes off, somebody... Oh, God. Somebody will have died and be donating their brain. Now, obviously, I got one of my staff to ring it on that occasion. But seriously, this is wonderful because it was you in the audience that suggested this last time. Quite a few of you said, you've tested us a great deal on medical and cognitive matters. Why haven't you asked to test our brains after we die? And so 173 of you have signed up to the brain tissue bank that we'll be talking about more after the break. And one person's family already has acted on the wishes of one of you to make sure that that happened. It happened just around Christmas, and it was done very effectively and efficiently, and the family were extremely pleased at this contribution that was being made. So the team is very, very grateful for that happening, and hopefully it won't happen very quickly. Right. The LBC 1921 tested numbers are as follows: 550 at age 79, 321 at age 83, 235 at age 87, 140 at age 90, and 59 at age 92, one of the oldest longitudinal cohorts that exists. What about some more numbers? I asked the database manager, Paul Redmond, to say, just how much do we know about the folks that were coming to meet this afternoon? And he said, look, don't even start putting in the genetics. Don't even put in the brain imaging. We have got 2,100 columns of information about you, not even counting genetics and brain imaging. Adele Taylor in the team calculated how many person-to-person hours of testing the team has undertaken since we first saw the first person in the 1921 cohort to the last person we've seen in the 1936 cohort. And she reckons it's about 21,528 hours of testing. We have joined with your information 45 international genetic consortia. So how famous are you now on the scientific stage in terms of scientific papers? So what about your science? You have been responsible for an astonishing 250 peer-reviewed scientific reports in international journals. That is absolutely amazing. 91 of those from the 1921 cohort only. I'm afraid the 36s have overtaken you in the last several months. The 1936 have been responsible for 118 papers, and led by Professor Wardlow, 43 of those papers have been on brain imaging, and 41 papers have been joint between the LBC 1921 and the LBC 1936. That's fantastic. Now, I dug out this slide from when I was talking to you in 2010, and we were pretty chuffed with ourselves at that point in having got to a stage where we're producing 11 and then 16 scientific papers a year. Look what the team is now doing. It's producing a scientific report of high quality in the peer-reviewed international literature more than one a fortnight, more than one every 2 weeks. Right. Now, can I just ask, is anybody from the press still here? Did somebody say yes? Okay, so the next stuff is embargoed, okay? So the material I'm going to show now is embargoed. It's not published, and I think you should be seeing some of it first. So some stuff that we have already published. You, the 21s and the 36s, were the very first to show, based on your DNA, how much genetics contributes to cognitive, mental function in older age. That only came out in 2011, and this is how many other research teams in the world have already cited this information from you. 180 times it's already been mentioned in the scientific literature. And that's what Professor Nick Martin said about that result that you produced. And that's astonishing from an internationally respected geneticist seeing that. And this article we produced in the world's top journal, Nature, in 2012, on your data, showing to what extent the environment and genes contribute to how much your cognitive function has changed over the years. It was read by a government minister who came up to Edinburgh to talk to us about it. David Willits, the Minister for Business, Information and Science, and that's what he said about the study. So you're well known, well cited by scientists, and even discussed round the cabinet table with Dave and his colleagues. And there's one of you here. That study attracted the whole of page 3 of the Times. Now, the story is very good. The headline is inaccurate. It was about cognitive change. It wasn't about dementia, as it says in the headline. Okay, sorry. Robin's asked me, could Mr. Miller please see one of the staff in the yellow jacket? Just follow Robin. Could Mr. Miller please follow Robin and see one of the staff in the yellow jacket? Thanks. Right. Now, these are some new and embargoed results. So again, for the press, these are not reportable. I think I saw Dr. Thomas back. Thomas, there he is, up the back with the bright orange jacket. Thomas is the type of chap that if he's got an idea in his head, he will come and annoy you about it. And Thomas started to bother me, and he said, Ian, why haven't you asked about whether people have attracted or learned a second language? I said, we can't ask them everything. But do you remember the questionnaire we sent you out? Well, Thomas has written this up now, haven't you, Thomas? And it's been accepted in a top American neurology journal. And it shows that, Thomas, correct me if I'm wrong, but broadly speaking, picking up a second or a subsequent language protects one from some amount of mental change or decline in older age. He's nodding, nodding reasonably well. So that's about to come out, and that will be covered, I'm sure, a great deal in the press. Now, this is another result we'll be sending off soon for publication. Do you all recognize that these are brains? These are actually your average brains, and I'm not stealing Professor Wardlaw's thunder because she's not going to mention this. But can you see any yellow or red on those brains? Okay. Those are the areas where the grey matter, you happy with that? Grey matter that thinks? Those are areas where the grey matter is thinner if you smoke. So we are the first really big study to show that smoking has an effect on thinning the grey matter on the outside of the brain. And that's a hot result we'll be sending off fairly soon. Right. One of our key ideas in the study. Now, you've heard me talk before about connections in the brain, haven't you? That's the white matter. The billions and billions of connections you've got in the brain. And part of the scanning that Professor Wardlaw does and we'll talk about is how good the connections of the brain are. And what you've been able to give us in your recent data is we've got 2 brain scans so we can study the change in the connections and the change in your thinking skills. And one of the key things we've been keen to study is whether people who have declined in the thinking skills are also declining in the connections of their brains. And we're about to be writing that up fairly soon. Right. Then I said, well, look, if there are 250 scientific reports, I can't possibly tell them everything that we've done. And so by way of getting you introduced to the research team, because it's the research team you should be meeting today, I'm just a talking head here telling you what other people have been doing. I did 2 things. One is that during the break, you'll be able to speak to them. One is that Professor Wardlaw will introduce you to the brain imaging team, but I asked members of the research team to pick their favourite paper from the 250 that we've produced to date. And I think it's quite interesting, the ones that they've chosen. So here we go. That's Alison Patty. And that's what she thought the most interesting paper that we've published is. What predicts tooth loss in the 1921 Lothian Birth-cohort? So John, well done with that. Alison thought that was her favourite paper. Alison is the longest serving. I think she's still got all her teeth, actually, Alison. Right. And that's Simon Cox at the end there. He thought the most interesting thing was in the Lothian Birth-cohort 1936, when we first found that brain connections, how well integrated your brain connections were related to how good your thinking skill was. Yeah, and Ricardo is around, and obviously we'll take you for a drink afterwards, sitting with a... Bloody hell, that's a gigantic gin and tonic, Ricardo. He's sitting with a huge gin and tonic, and his favourite thing was this: alcohol consumption and lifetime change in cognitive ability. Oh, God, this is a hard one to summarise. It basically shows this. If you've got a certain type of genes, it will boost your cognitive function if you drink, and if you've got the other type, it will decline. So you don't know what it is. Just carry on. Sorry. Dave Leewald, I'm sure all of you have this tripping off your tongue. Common variants, a large effect in F12, KNG1, and HRD are associated with activated partial thromboplastin time. Well, I think I know why Dave picked this, because this was the very first study we published based on the hundreds of thousands of genetic tests that we did on you. And we couldn't believe we had discovered this, a contribution to clotting, blood clotting time, before anybody else had discovered it. And we got in first with this. I think that's why Dave, and Dave chewed all the file processing in this. So Dave's our computer programmer in the team. Ah, yes. So Stuart Ritchie, you can see he's a keen young man from the photograph there. He's a bright spark. And he thinks that the best paper was another 1921 one, Height and Intelligence in the Lothian Birth-cohort 1921, a longitudinal study. Stuart Ritchie, 6'0". So he's pumping himself up a bit there as well. That's Caroline, who many of you will know, Caroline Brett. Her favourite one was that those of you who are more active have got less brain shrinkage and got better connections in the brain. She published recently. And Gail Davies there, her one that she picked was the one that David Willits came to see, and a quotation there as well. Now, Gail might be seen to be a bit of a sook here because she's picked a paper that I authored first as well. So you have to take that with a pinch of salt. And also, she's given a picture there. She's not exactly name-dropping, but that, who knows actually who's sitting with her there? Because I think I talked about him last time. That's James Watson of Watson and Crick, who discovered the structure of DNA, and he came to visit the lab in 2010. And Janie, who many, many of you will know, liked best that childhood cognitive ability contributes to the thickness of the outside of the brain. So Janie says there are advantages to being thick-headed. She means thick-brained, but we'll live with that. Very nice picture. And the same study was picked by this big, handsome hunk of a man, David Dickey, one of our brain imaging processors as well, and he liked that paper too. And Sarah Harris, who's one of our genetics workers, liked the study that found a particular gene that if you've got one flavour or another contributes to how strong your thinking skills are in older age. Now, I think that's been nice. We did not choreograph any of that. We just got people to pick what study they like, but it gives you a flavour of the sorts of things that you're contributing to. And Michelle, I thought I'd leave this one till last. Have a look at the title. Ah, you're laughing now. What we did there was we got 10 students to rate every picture in the Lothian Birth-cohort 1921 and ask the question, how old does this person look? They didn't know they were all the same age. And we followed that up, seriously, over the next 10 to 12 years. And how old those students thought those people looked was a stronger predictor of their actual age, of how long they lived. So that was an interesting one. And Paul Redmond, who's wearing fewer clothes there than I'm happy with on that photograph, thought there's more to life than thinking. And so he liked the study that was funded by an Age UK PhD student that looked at things other than just cognition, to do with physical aging, emotional aging, and well-being. So we're interested in well-being other than just thinking skills. Right, I'm just going to finish with a few numbers to give you some results of the data that you've just given us. Right, let me just check this. I just want to check that, yeah, they're all been updated. Okay, right. What I'm going to do is now remind you of the test you took. Now, do you remember I did this last time? Sorry, it's not a test, but I gave this to you last time. Now, do you remember that story you've got to remember? Well, you're nodding. There's actually two, okay? And if any of you mentions the name of that woman that's robbed, I will kill you because, okay? Shush. Right. How well did you do? So can you get your eye into this slide? Because you're going to see a lot like this. So age 70, age 73, age 76. So here's the score. I think we'll just shut the study down now. I'm not happy with you lot. You're supposed to be helping us study cognitive aging. Okay. Now, I'm going to show the 21s, but the 21s are scored in a different way, so they can only be compared with themselves. That's blooming useless. Okay. Remember that test? Do you remember the backwards one? Where you've got to point to the blocks backwards? You're just as good at it at 76 as at 70. Do you remember that one? You've got to rearrange. I don't like doing that one, actually. You've got to rearrange the numbers and give them in numerical order, then the letters in alphabetical order. Hear how you're doing. So it's gone down a wee bit, but not much at all. Do you remember the one where we give you certain letters and you've got to find as many words as you can? Not much happening there, is there? Let's look at the 21s. So you're going to do pretty well on that until your 90s. Do you remember the one where you get these pronounced words? Can you see I'm going to give you all the good news first, by the way? Okay, so that's the average score at 70, 73, and 76. Here's the 1921s. They have taken this test 4 times. Look, they're doing better than the 36s. Do you remember the one where you've got to... what was that face for? You don't like that one? That's because you're having to think a bit instead of just tell us words that you know. Anyway, how do you do on it? So it's gone down a wee bit. Is this clear to everybody what these are showing? These are the average scores as we go through the age. Now, again, I'm going to stress the 1921s get a different test, but they do the same sort of thing. But can you see that they do go down on 21 from age 79 through 83, 87, 90, and 92? Is that clear? So working out these rules and applying them to these patterns goes down a wee bit. Do you remember the blocks? Actually, it's quite good, actually, these reactions to the individual tests. I can probably tell what they mean. Anyway, these are good fun. And how well do these do over time? So age 70, age 73, and age 76. So slipping just a little bit. Remember, you've got to write in these codes as fast as you can. So what I'm coming to now are the tests that are primarily to do with speed of thinking. One of the main things that we're interested in. So here's the score. And watch as we go to the 1921s, who took this 4 times, beginning at age 83. So again, just to go back. That does go down a bit. Do you remember you had to press a button as soon as you saw a 0? That's a simple bit. Now, wait a minute. So we've come to the first one where a high number is bad. So you want to be going down. You want to be going down. So that took about 1/4 of a second. That is 270 thousandths of a second. So you can all work out 250 would be 1/4. How's that doing? Well, it goes up a wee bit. What about do you remember the one where you've got to do 4 fingers? You've got to make a choice. That takes longer. So at 70, you were taking, what is that? Just under 2/3 of a second. 630 thousandths of a second. How does that do with age? Now, remember that last number, because the 1921s were good enough to help us with this test as well. So that slows more markedly than anything else we've been finding. Now, one of my favourite tests, but I'm expecting a big groan when I show you it next, is this one. Yeah, exactly. It's where you have to stare at a computer screen for 20 minutes, and it is a very good test, but I know that not everybody likes doing it. How do you score on that? Higher numbers are better. Higher numbers are better. So, slipping a wee bit, but not very much. Right, let's go now to the test you originally all took. This is what you all share here, those of you who sat the test at age 11. See how well you scored. So that's the average score, 65.5 at age 70. You didn't take it when you were 73. Right, I want you to keep that in mind because we're going to move on to the 1921s now. So, almost identical. But what I'm going to show you now is what they scored when they were 11. Can you see? So even when you get into your 90s, you're still not getting down to the score that you were at age 11. Right. If I do a graph of the scores that are to do with speed, can you all see they are going down over time? But that's the average, and you're all individuals. Do you want to see what happens if I put individual scores on these over time? That looks quite clean, doesn't it? This is what Stuart Ritchie gave me. So, although I can tell a clean story with averages, can you see that some people go up, some people stay the same, and some go down? Look! It's much, much, much more about the individuals. And what I'm trying to do in this project with my colleagues here is study this. Why the mean does not tell you the full story. What we are trying to understand is why people are diverging so much as they grow into these ages and 70s and 80s. So I'm going to finish with just say that other than the science, that's some of the new science, please talk to the team during the break about this. But in addition to this, you know as well, you've become highly attractive, not just to scientists, but to artists. A lot of you came to the play that was produced at the festival a few years ago, Still Life Dreaming. Some of you came to see the exhibition of photographs and videos that Linda made, that was its own exhibition. We've actually got Fiona Carlisle here today, the renowned portrait artist who, Fiona, you've done the only official portrait, I've just caught her eye, of Alex Salmond. Yes, so Fiona's done the only official portrait to date of Alex Salmond. And here she's done one of our 1921 cohort. And here she is painting some old bloke in the research team. And the professional writer Anne Lingard has just published a book doing the stories of some of the individuals in the team. And that'll be available today for you. We represented your research at the House of Lords summer science reception as well and told them all about the findings. John Starr and I have contributed to Age UK's books to help older people learn about ageing well, 2 of them now. You've contributed to the University of Edinburgh drawing in a medical research council centre for cognitive ageing and cognitive epidemiology that's brought 10 new staff to the university. You've contributed to Professor Starr's bringing in from Alzheimer's Scotland a dementia research centre that partners a centre above there in the department. And that's what I've got to say. So really, it's just a way of saying we value what you contribute very much. We think it's important to have these reunions, partly to ask you to come back, but also to show you the results before anybody else gets to see them. And thank you very, very much for your contributions. Thank you. And now for much more interesting and scientific material, I'm going to introduce 2 of my colleagues. First of all, Professor Joanna Wardlaw is going to talk about the amazing work on brain imaging on a group of you who've contributed a number of brain images that's relatively rare across the world in terms of its number and quality. And then I'll introduce John Starr to talk about the medical. So, Professor Wardlaw on brain imaging. Joanna.
Thank you. Well, thank you very much, Ian, and I can't emphasize enough what an honor it is to be able to work on this project. The information that you've contributed is absolutely unique, and it's really contributing to major understanding, major advances in how we understand both normal things as well as common diseases as well. So I'm going to say just a few words about some of the work that we've been doing to update you on the brain imaging. And thank you very much to the many of you who came and had a brain scan. You'll remember this machine. It's the loud, noisy one that's like going into a tunnel. Despite its sensation, it's very safe and it's extremely boring. And I frequently fall asleep in there. It's the one place where they can't get you on the phone. So it's one way of escaping. But it's a very powerful tool, and it enables us to collect all the information on wiring in the brain and structure in the brain and all sorts of other things which Ian has mentioned, and I'll show you a few examples of what we've been finding out. What we're trying to look at is things like, why does the brain change with age? We know that as well as changes in your thinking skills, that your brain gradually gets smaller as you get older, and this happens to everybody. And we don't really understand what drives that and why some people do that more than others. So that's one of the things that we're trying to find out. And also particularly, is there anything that we can do? Are there strategies that you can do? Are there simple things like exercise, which seems to be one of the things that's coming through, which can help stop that process? Because we know that the smaller the brain gets, it also tends to be associated with the thinking not being as good as it was. Other things that happen in your brain that change as you get older, and again, this is something that happens to a certain extent to everybody, are that you develop these areas of these white splashes in the brain, which we don't really understand what causes them. But one of the very striking things is that for a long time, they were seen on scans, and everybody just thought they were part of normal aging, a bit like wrinkles. But of course, everybody gets them to a different extent. When you're in your 50s to sort of late 60s, about 7% of the population have them. But by the time you're in your 70s to late 80s, almost a fifth of the population will have them on average. But some people have more than others, and we know that they affect your thinking skills. So it's important to find out, they're not just something which is an automatic accompaniment of aging. So again, how can we stop this happening? Why does it happen? How much happens? And then the other thing that Ian mentioned, and you'll be aware of, is this business of trying to study the wiring in the brain, the connections in the brain. And the part of the scan where the scanner gets particularly noisy is when it's collecting this piece of information. And you may remember from previous presentations that we can use the information from the scan to look at the way the wires are running in the brain, your white matter pathways, and we can use the information to construct mathematically a model of what that would look like. And when we first started doing this, my colleague, Dr. Mark Baston, who's a very clever medical physicist who's done all of this work, was very good at putting together individual pathways and being able to follow just individual pathways. And we spent a lot of time working out really good ways of doing this. But now we're beginning to produce what I would say are absolutely fabulous images, not just of single individual pathways, but of all the pathways in the brain. And here's just one example of that. And once you've got to that stage, you're almost painting the wiring in the brain. It's really quite fantastic what you can do with it in terms of linking it to understanding about those cognitive tests that Ian was showing, studying little areas and how those different areas affect things. And then also you'll remember the test on the neck where there's a bit of wet jelly and a little camera held against the neck, which is looking with ultrasound, same type of test as we use for scanning mothers when they're pregnant, looking at the blood flow into the brain, so the energy that's getting into the brain, the oxygen, also looking at how well the blood vessels are staying open, or are they getting more furred up with the sort of fatty deposits that tend to happen in our society, and how stiff are the blood vessels getting. So there's several things that can happen. All again are part of aging, but they happen to different degrees in different people. And one of the particularly unique things about this study is that because we've got both the brain imaging and the information about blood flow into the brain, we've been able to do some fairly nifty analyses about how these two things relate to each other and to things like cognition. It's important that the blood vessels stay wide open. Your brain is a very important organ, but as you can see on the bottom, that's what happens when the blood vessels get narrow, and then of course it's like your garden hose getting a kink in it. Not as much blood gets up there, and your plants or your brain doesn't get as well watered as it should do. And the team. So we've got a large team, and many people involved in this project have also done other types of work. And some of the people that were employed when we first started doing the brain imaging have moved on to doing other things. And this is just a little summary of everybody. There are a number of radiographers that do the actual scans. You'll remember meeting them when you come for scanning. When we first started, we had 2 people who worked on processing all of the images, Susana Maniega and Maria Hernandez, who just by chance both happen to be Spanish by background. Susana is still working on the project, but Maria has moved on to a lecturing post and promoted. We've had other people who've come in for a short time, and they were so clever that they've gone on to become professors in other departments. Several people have got a PhD, and they've gone on to other work. And we're pleased to welcome some new people to the project, including, as Ian has already mentioned, David Dickey, who's now working on processing all the image data to get the most accurate information out of it that we possibly can. So, very briefly, what sort of things have we found since we last met? I've just picked out a few highlights. As Ian said, we have quite a large number of papers, and they're coming out faster and faster, and the world is really beginning to learn a great deal from the information that we're finding from you. So we mentioned about the white matter in the brain is really important, but we're also interested in the grey matter with little grey cells that lie over the surface of the brain. And for up until now, there's been a great deal of interest in the grey matter in the brain, and people thinking that that's the most important bit. And we know that the grey matter gets thinner as you get older as part of the general shrinkage of the brain. But because most studies look at people just when they're older, they don't think about what they might have been like when they were younger, it's always been assumed that the thinning of the brain when you're older is just because you're older, not necessarily because actually that's the way you were to start with. And of course, it's really important that you consider how you were to start with. Otherwise, you'll assume that things are just due to aging or just due to something else, when in fact they're not. So you've already seen a picture that looked a little bit like this, except the colours were all in yellow from Ian. This is from a similar analysis where we looked at the thickness of that grey matter over the surface of the brain. And this is what the difference is. So this is saying how much of the thickness of the brain in old age is because of age, and how much of it is because of, actually that's what you were like when you were younger. And so when we correct for your tests that you did when you were 11, we found that most of that apparent change with aging is actually because that's what you were like when you were younger. So immediately it means that we are able to study much more accurately the amount of thinning or the amount of shrinkage which is just due to being older, and not what was there all along right from the very beginning. And that's really, really important. Nobody's actually been able to do that properly before. So what about the white matter? Well, I mentioned about these white splashes that appear in the brain, and we know from many, many, many studies, not just this one, that the more of this damage that you have, the more it affects your ability to think, mostly speed of thinking, and also things like movement. It's not just about thinking, it's about general functions as well. But we're not quite sure why. Many people have thought up until now that it was all just because of things like high blood pressure or smoking or being diabetic or eating too much cholesterol in your diet and that kind of thing, because we just tend to blame those things for everything. But some of the analyses that we've done have shown that yes, of course those things are important, but they only explain a very, very tiny, tiny amount of that white matter damage. Actually, about 98%, most of that white matter damage is not explained by those common things. So what we're starting to do now is drill down into what does actually explain those changes. We know, because you've had these tests when you were 11, that your thinking ability at the age of 11 influences how much of this white matter change you get when you're older. And that's really interesting, because what it's starting to suggest is that if we want to stop this happening, we actually have to not just think about doing things when people are in their 50s or 60s or 70s, we actually have to think about things like school policy or other, society policies. We know that genetics is probably important and that several little differences may add up to cause more problems, but it only also explains a very small part of this, and that it's partly about the wiring, it's partly about how you handle energy in your cells, all sorts of things. It's not a straightforward, simple thing. So here's one of our other very fancy images where we're now starting not just to look at the overall damage, but we're looking down in detail at the wiring. And this is an image that shows not just the wiring, but how those areas of damage start to affect the wiring. So you can see these large blue areas here. Those are the areas of this white matter damage. And what we're starting to study now is how those areas of white matter damage actually affect the wiring and influence how it can transmit signals. We know that the wiring is really important for your thinking skills. And we also know that it's partly genetic, purely because of the information that has been provided from this work. We know that things like smoking and blood pressure particularly increase the amount of damage. And as I've said, they only account for a small bit of it, but it's still really important. And interestingly, it looks like things like exercise might actually help stop that damage. And some of the other intriguing observations are that if you've got good wiring, that may actually help protect you against getting damage in future. So that's where this shift from thinking about things that are just happening to you as we all get older, but looking back into what is it about the way you're set up when you're younger that influences these things is becoming really important. So my final couple of slides, we're starting to home in on what is this actual damage, because if you really want to do something about it, you have to understand what it is that's causing it. And we've done a lot of, when you have your scan, you get several different scans, and each one of those is telling us something different about what's going on in the brain. And we've used those in combination. So each one of these little tiny pictures, little stamps, shows a different type of image, information that we get out of the brain scan. And some of this tells us about things like how much water is there in the brain. Some of it tells us about things like how much of fat that insulates the wires is in the brain, all sorts of things like that. And basically, when you put all of that together and you say, what is it that seems to start to change first that then leads to these white matter damage areas happening, the very simple thing is that it's more water. There's more water in the brain than when you have less of these changes. Now, from older studies in the past, looking at things like pathology tissue, the brain after people have died, when the changes are all very late, people were assuming that it was all because the actual wires were falling apart and the coating on the wires, the fatty coating, was being destroyed. What this says is that there's a series of stages before you ever get to that point where a lot of early subtle things are happening, which could be much easier to try and do something about. And it's all about water, not about fat. So my final slide is to emphasize how important it is to have the information from all of you normal people. Because as Ian has said, there is almost no such thing as normal. Everybody's a little bit different, and capturing that range of normality is really, really important, not just for saying what is normal, but also for saying when does somebody start to become abnormal. Because if we want to be able to improve the diagnosis of things like dementia or things like stroke in future, we need to be able to define those boundaries much more clearly. And this is some work which David Dickey did as part of his PhD project, but which we're hoping to continue now that he's working on the LBC cohort data, showing that there's actually a much wider range of variation in people who are normal than has previously been thought. And equally, once somebody actually has a diagnosis of dementia, and this is not from you, this is from another data set somewhere else, there's also quite a wide range. And so if you just try and say, okay, once this bit of brain gets to a certain size, then that's abnormal and that's it, you can't actually do that. You have to have a much wider understanding of what the boundaries of normal are to be able to do that. And we're building up gradually all of the data that we've got to be able to create atlases that say, what are the boundaries of normality, and it's like contours on a mountain, and when does somebody actually become definitively abnormal, and when is it just within normal limits, but still normal. Okay, so a bit of challenge for the future. We're still trying to work out how fast these things happen, which is why looking at scans over a period of time at the same time as having cognitive tests over time and all the other tests that you do is really, really important, because you can't just look at one of these things in isolation. You have to look at all of them together. And we're in the process of analysing all of the data from the scans that you most recently had so that we can look at these changes. If we want to be able to do something about it, then we need to understand what things might be changeable. And the genes that you're born with are not really something that we can do much about. But we can do something about how you live, what you eat, how you choose to, spend your spare time. So how much of it is due to environment is one of the big interests of this project. And how much of environmental things influence your brain is very much part of what we're doing. What causes the changes in the first place? Can they be reversed? I promised I wouldn't crack any bad jokes this time, like I did last time. But I was just tempted to mention that some of our recent analyses were interpreted as suggesting that your brain might actually get bigger. Ian and I are still arguing about whether that's possible or not, but it's one of the things that we're certainly interested in looking at. And then, does the prevention of things that really, when aging is not successful, and when people get disease like stroke or dementia, or other common brain diseases that are associated with aging, do we actually need to start thinking about strategies which would enable us to help prevent those things happening, really starting right back in youth? Because some of the information that we're getting now does suggest that yes, of course you have to treat blood pressure and make sure that your diet is good and that you get exercise, and please, if you can possibly stop smoking, it's a good idea. But there's other things, and maybe we need to have policies that just move things earlier and be more aware of important societal things that could help to keep the population healthier for longer. And this is a completely unique opportunity to sort this out. And I would just echo Ian's comment. We really hope that you will come back for further testing, all sorts of the testing, not just the scanning, although I really hope you'll come back for the scanning. And we would very much like also to look at the blood vessels in the neck again so that we can see how those things have changed as well. And the longer we can go on doing this, the better. And it's an absolutely unique opportunity, and it's been so valuable even now, but it could be even more valuable if we can carry on. So thank you very much.
Be question time afterwards. We've got to bash on because some people have got to leave at the break, but note it down, okay? And we'll answer it then. So, oh right, okay. Are you the one that examines the brain scans? Yes. Yes. Okay, right. She's flying off somewhere actually, so you better be fast. Yeah, look forward to that, Joanna. Right. So we did some things right. It turned out the reason that you've been so successful internationally is that it's very unusual to know about the mental test that you took as children. That's turned out to be incredibly valuable. We were right to include people born in a single year, 1921, 1936, because it means that there's no variation in that year. And it was right to do repeated testing time after time. Those are the things that have led to you being so successful. But Professor Wardlaw and I have mostly talked about mental tests and functions and brain scans. But we're going to move away to the rest of health now. And Professor John Starr is going to talk about health.
Oh, what a delight it is to be here. And to be honest with you, it's like Easter come early when you get your hands on the data. Last week, I got my hands for the first time on your data to tell me what's been going on. So isn't that fantastic? It's like opening lots of Easter eggs. So I want to tell you about it today. I haven't had a chance to open all the Easter eggs, so I've just made a selected few about health. And you'll remember when Ian presented that he was saying about how some things cognitively didn't change, like how you pronounce words and then some things to do with speed did change. And you'll see a similar pattern coming out, so that's quite of interest, particularly listening to Joanna about all the wiring in the brain and how it relates to speed of doing things, speed of thinking, speed of moving. Okay, so how are things doing? Well, in the general picture, I thought I'd put this graph up first. I'm going to show you a lot of graphs, and this is the graph of the FTSE 100. I've looked at the FTSE 100 on the 17th of April over the last few years, from 2007 to 2013, essentially every April the 17th, except sometimes it was a weekend, so I had to take a little day either way to see what's been going on. And here it is. And guess what? The FTSE today is about the same as it was in 2007. Nothing's changed. And if you look at the graph, it went down a bit, then it went down a lot, and came up a bit. And it just shows you things can go up on average or down on average. I'm going to present some averages, but you'll remember within the FTSE, which is the 100 companies that are listed, that some go up and some go down. So if you'd had your shares in Royal Bank of Scotland, you wouldn't be doing quite so well as the average people over the last 7 years. Okay, so let's have a look and see how we've been doing. And you were I was very pleased to see that height came as one of those favourite papers. People are interested in height. Everybody's interested in height. And there's this idea that we lose height over time. And certainly that paper we published for the people on the LBC 1921s showed they did lose quite a bit of height, but not until they were in their 80s. So people born in 1936, how are you doing? Over the last 6 years, how much height have you lost? And there it is, very, very little. On average, you've lost half an inch over 6 years. That's not bad. And that means some people actually might be standing up a bit straighter. Some people might be a bit more crouched. But on average, just half an inch. And that doesn't matter whether you're a man or a woman. You've lost the same amount. Okay? So this idea that people kind of shrink as they get older, well, maybe, but not very much. So you don't have to buy out a whole lot of new clothes because they're still going to fit you. Well, maybe you do need to buy a whole lot of new clothes because maybe you've put a lot of weight on, or maybe you've lost a lot of weight. What do you reckon? Shall we have a look? Here we are. Weight. 1 pound on average. Now, how can you do better than that? You haven't changed. Your height hasn't changed. Your weight hasn't changed. You could be wearing the same clothes now as you were back in 2007. Isn't that fantastic? That's brilliant. Some things don't change. Nobody's been on big diet. Well, some people might have been on a big diet and some people might have put a few pounds on here and there. But that just shows you, in general health terms, weight is a fantastic indicator of how well you're doing. Now, this is a selected group because obviously we only weigh the people who come back to be tested. There will be people who don't come back who may not be doing so well, and they may have lost some weight. They may have put on a lot of weight for various reasons. So this is a selected group. This is just these are just the results, the average results of you sitting here and a few other people who can't make it here today, the people who came back to be tested on this last wave. But if you got back, if you were fit enough to get back, and maybe certainly if you were fit enough to get up these stairs today, you can be sure that on average, your weight and your height, absolutely steady. You're the same person now, well, apart from the fact your cells have all changed over and over and over again, but you're the same person now essentially as you were then. Isn't that fantastic? How do we keep that the same? That's a really interesting question, isn't it? How we stay exactly the same over those number of years when all the other things are changing. Okay, so that mirrors some of the aspects of your mental abilities, the memory side and the pronunciation side. It's amazing how things... So this is not a story completely of change. This is partly a story of things being the same. Yeah? Things enduring and being resilient. Okay. So what has changed? Look, 2007, here we are. They're here now, aren't they? They're here, but we're not actually able to get on them, but, they're here. And what reminds me is that here's a picture of some people walking off the tram. And one of the things we get you to do is to see how quickly you can walk along. Not for very long. You don't have to walk, miles and miles, just a few meters. And we see how fast you can walk. And you had to walk up the stairs today. And I don't know if you found it a bit more tricky now than you did last time you came up the stairs. Were you going a bit quicker up the stairs, a bit slower up the stairs, much the same? I don't know. But we do measure you. And this is something where there has been a change. And you'll relate this to what Ian was saying, what Joanna was saying. That when we measure how fast you walk, over the piece, whether you're a man or a woman, you've slowed down by 25% over the last 6 to 7 years. That's quite a big change, isn't it? I should say that that's a proportional change. So actually, in absolute terms of miles per hour, women have actually slowed down a bit less than men. So men aren't striding quite so far ahead down the pavement as their wives now, you know, because men and women are women are catching up the men. But there has been a change, and that's quite a significant change in your 70s. And that's something you may or may not have noticed, I don't know. But that's certainly true. So even though you're the same height, your same weight, so you're not kind of carrying any more weight, your legs are just as long, you're going a bit slower. And maybe that's because you're not quite so strong. And you have lost a bit of strength, a little bit of strength, not a lot. This is when you squeeze that handheld, what we call a dynamometer, measures your power. So this is when we get you to grip hard as you can. You've lost a bit. You were doing quite well, 70, 73. You've gone down a bit at 76. This is the bit that affects when you can open the jar and things like that. Again, women, although they're not as strong as men, are actually keeping their strength up a little bit more than men. So actually, women are doing a bit better, relatively, than the men on this. Okay, so they're kind of catching up. So, but that doesn't really explain the change in walking speed. You know, there's a bit of change here, but it's not enough to explain the change in walking speed. It's not because you've suddenly become very weak at the knees. That's not why you're slowing down. Okay. But these things do have an effect on everyday life. Here, I've put a few pictures up to remind me of the questions that we ask you about your everyday function. Things like, can you carry heavy shopping? Can you get on and off a bus? You remember that test that we asked you? Just a few questions? Only about 9 questions, but they are really telling about how you can manage in everyday life. And they relate to walking and grip strength, etc. So let's see how that's going. So this is what I call the activities of daily living impairment. And here, the higher the score, the worse things are. This is scored out of 18, all right? So the worst you could be is 18. So at the very beginning, and even now, the scores are very, very low. You're not in any way highly impeded in terms of you can still do those things, reaching up to a shelf, tying a knot, getting on and off a bus. You can do nearly all of those things. You could do nearly all of them things to start off with. So for women, there was just one item on average they could do that needed a little bit of assistance with. But now, there's just a bit more assistance needed. Maybe because you're not able to walk quite so quickly or do things quite so sprightly. Just a little bit is changing. Just a little bit more impeded in the kind of everyday life. It's not very noticeable. You're nowhere near having major problems. Just a little bit. It's the area where we, doctors would look at it as an area for what we call prehabilitation, doing something before things start to get down into the more high levels, the 6s, the 7s on this score. It's an area where we can see some change. It's an area where we could start to do things. Okay, so that's important. It shows you that some of those changes, like how fast you walk and things, are related to how your everyday life, carrying the heavy shopping, etc. What about changes in health? So we kind of see from you, we ask you what's happened to you over the last few years, and I haven't put down the baseline here, because there's obviously a proportion of people who already have diabetes. There's a good proportion of people who have high blood pressure, hypertension, and there's a good proportion of people who'd had some history of cancer at some point in their lives. What has happened over the last 6 years? So, 5% of people who didn't have diabetes have developed diabetes. So, about 1% a year. It's not big, but it's up there. What about high blood pressure? GPs must be very keen on this. Perhaps they're getting some money for diagnosing it, I don't know. But nearly a third of you, over 6 years, certainly between a quarter and a third, have got a new diagnosis of hypertension, high blood pressure. So that's a major thing being treated. Now, there's lots of guidelines, and it's a good thing. We've heard Joanna talk about the importance of blood pressure. But it just shows you how many people are starting to get these kind of conditions. It's difficult to call it an illness, but these conditions as you get a little older. What about cancer? 14% of people who didn't have cancer have had cancer over the last 6 years. Okay? And that's the people who came back. That's not the people who didn't come back, because obviously there'll be a proportion of people who got cancer who didn't come back for testing, for various reasons. So probably the figure's a little bit higher than that. So that's a major issue. So if you looked at health, the health changes for you in your 70s, between the age of 70, 76, 77 now, 78 as we're coming on to, there's big changes, quite big changes. Big changes in health, your weight staying steady, your height staying steady, but there are changes in health, very small perhaps impacts in terms of how you're managing in everyday life, a bit bigger in terms of how quickly you can do things, but big changes in terms of the health profile, how much you're having to go to the doctor, what kind of tablets you're taking, visits to the hospital. So, here's the summary. Almost ready to hand over for the film and then off to tea. So, height and weight is steady. So that's good news. Everything, there's resilience and endurance. A bit fit, a bit less fit, a bit less strong. And that's possibly just starting to affect everyday life. But perhaps the biggest difference that we're seeing is the increase in amount of disease. It's not necessarily making you less able to do things in life, but certainly more visits to the doctor, more visits to hospital. And, well, we shall find out over the next few years how that continues. Thank you very much.
Thanks, John. Now, we've got some friends from Age UK who've come up from London today, and probably they're all wondering why John got a big laugh when he showed the tram. And the answer is £700 million to replace a perfectly good airport bus. On. Right, if that's what gets on the news tonight, I'll blame you lot. Right, we've had the update, and we're going to do something unusual. We're going to show you an exclusive preview of a professional film that's been made on the Lothian birth cohorts. Now, it's not a documentary describing all about them. It's a professional filmmaker and artist's reflection on what it means to be in the cohort. But I thought too, because some of our colleagues from Age UK have to leave, this is a good time to thank people. So I'm going to do a quick thanks. And if I've forgotten people, I apologise. I don't usually read from a list, but this time it's so long I need to. First of all, the Scottish Council for Research and Education did that test at age 11 and allowed us to use the data. Thanks to the participants, you in the 1921s and the 1936, my excellent co-principal investigators, the research team who've done all the work that we brought to you today, particularly Joanna's mentioned the imaging team, but in the LBC team that you know just now, it's Alison, Janie, Adele, Simon, Paul, Stuart that you'll meet later between now and the second half in the break time. The Wellcome Trust Clinical Research Facility nurses and administrative staff. Lee Murphy and his team at the Wellcome Trust Clinical Research Facility Genetics Core. Of course, the neuroradiology team that Joanna's mentioned already. The Doppler ultrasound scanning team. The team at the Centre for Cognitive Ageing and Cognitive Epidemiology. Our fantastic collaborators at University of Edinburgh and those Firth of Edinburgh, and funders BBSRC, Medical Research Council, Lifelong Health and Wellbeing, the Scottish Government's Chief Scientist Office, the Wellcome Trust and the Royal Society. But most of all, and I'm leaving it till last, the major funders for the Lothian Birth Cohort 1936 for 10 years now, and to the tune of several millions of pounds, are our friends sitting up the back. And just to embarrass them, let's all look round at them and thank them very, very much for their funding for the 1936. Only distinguishable by their current embarrassment. And Tom Wright, who's the Chief Executive Officer of Age UK, who Libby is currently pointing at, came up and spent the morning with us and has enjoyed being here today to see actually some of you here because he's heard so much about the Lothian birth cohorts. So we're going to see you after the break for your questions, please. And Simon Cox, our study coordinator, is going to give you some information. And you're not looking at me because I should have said something else, are you? No, come on, Simon, tell them how to get their tea and what we're doing in the next wee while. Hello, everybody.
So Ian also asked me to give you a very, very quick overview of the work I do as a scientist on the LBC 1936 study. So I promise to keep it brief. I know you're all champing at the bit for a cup of tea. So I'm really interested in your brains, tremendously interested in them, particularly in the different compartments of the brain. It seems that different parts of the brain are specialized to a certain degree for different types of thinking skill. And so what I'm very interested in doing is understanding not only how we can measure these bits of the brain, but how they interact with other areas through the connections that Professor Wardle's already talked about. So I'm interested in how these different networks that are formed within the brain are composed, how we actually can measure them from the detailed information that you've given us in your brain scans, how they change with age, and what are the causes and effects of this. So what does it mean when the composition of these brain networks alter as we get older for your thinking skills? And also, what kind of things might be driving these changes? And it's really worthwhile mentioning here that actually this set of questions is a set of questions that probably couldn't be answered in any other data set all in one go than in the LBC 1936. So it's a really rare opportunity for somebody with my particular interests to look at this. But when I'm not doing that, I also coordinate the LBC 1936 as well. So time for a bit of coordination. And this is all to help you get your cups of tea. Before we do that, however, what I'd like to do, if possible, is have all the cohort members, so the 1921s and 36s, to come out through the side doors here along with everybody else, but to go down to the front steps so we can get an updated version of that really iconic photograph of you all on the front steps. So, in fact, our friendly neighborhood photographer here is going to get everything sorted out, so if you'd like to follow him when you're on the front steps, he'll give you directions. Following that, tea, biscuits, and coffee will be available in 2 different places. So we don't have one room which can fit you all in, unfortunately. So if you're sat to my left here, when you come back in again from the photograph on the steps, I'd like you to try and turn left, and colleagues of mine on the team will direct you towards the tea and coffee, which is actually being served in the gallery behind there. Then when you've finished, come back through here. You'll feel free to circulate, bring your cup of tea and coffee back into this room here. If you're sitting in the middle or to the right, when you come back in from the photograph, turn off to the right, and there's another room over there that my colleagues will direct you to. A couple of quick other things to look out for during the coffee and tea break, which will probably last around half an hour or so. We'll have you back in here at 4 o'clock. First of all, as Professor Deary's mentioned, we'd like to get your feedback and questions for the Lothian birth cohort study. So there's a couple of boxes just out at the front there with lots of bits of paper and pencil. So please do jot down a question if you have it, and I'll fire them at Professors Deary and Starr at the very end of the session today. Also, let us know what you think are the best days of our lives. Again, that's for Professor Deary's debate at the British Academy, which is going to be taking place on the 29th of this month. There's also a small selection of class photos provided by members of the cohort along this corridor here to my left. So feel free to have a wander along and see if you can spot yourself or other people that you might know. Also, team members, some of whom you'll recognise, such as Alison, Janie and Adele, who are along the top of this slide here. But also other members of the team who don't get to see you very much will be standing next to posters in this black and white corridor at the front here. And we'll be happy to speak to you, so feel free to collar them and discuss the kind of research that they do with your data. Finally, meet the artists. So you've already heard a lot about the portraits that Fiona Carlyle has done already on some of the cohort members. The LBC Lifetimes book that Anne Lingard has written about LBC members and scientists and their experiences in the study and beyond. And as well, we're going to be giving out copies of that book to you all at the end as you leave at 5 o'clock. And also Anne Milne, who made the film that you just all watched, and they'll all be here today, so feel free to look out for them as well. Okay, so that's the end of the first half. As I mentioned, if you can now leave in an orderly fashion through all the side doors, and colleagues will direct you down to the front steps for the photograph. Thanks very much.
So before the questions, we're going to have 4 quick fire talks from 4 young women. and there are things that I hope you find particularly interesting. So each going to speak for 5 minutes. And first one is Philippa Dahl from Glasgow Caledonian University. Now, we are hoping in the team to start testing again for wave 4 of the Lothian Birth-cohort of 1936 from late this autumn. And it'll be the same again, medical and cognitive testing. And we'll invite you to retake the brain scan. But also, in addition, you will be offered the opportunity, you don't have to do it, you don't have to do anything, but you'll be offered the opportunity, as will all my staff and me, to wear this tiny little thing for a week, strapped to your leg to see how much you move around. And I'm not going to say any more about that, but I'm going to do it. I'm going to make sure the team all do it, and Philippa Dahl is going to tell you something about it on the project called Seniors USP. Philippa, do you want to come and say?
Hello. As Ian has already said, I'm Philippa Dahl and I work at Glasgow Caledonian University. And I'm here to talk to you about the Seniors USP project. And this project is a collaboration between researchers in Edinburgh and Glasgow and is being funded by the Medical Research Council. And we'll be working with the 1936 Lothian Birth-cohort as part of the next wave. So Seniors USP, as you can hopefully read in the small writing up on the slide, USP stands for Understanding Sedentary Patterns. And the project is all about measuring and understanding sedentary behaviour of older adults. Sedentary behaviour is basically sitting and lying down while you're awake. So we might have been better calling the... Yes, it's ironic, I know. We might have been better calling our study Understanding Sitting Patterns. So you might all be there, sitting down, looking at me, saying, why on earth are you interested in how much people sit? Well, unfortunately, sitting for prolonged periods has recently been found to be a risk factor for poor health. And when I talk about sedentary behaviour, I don't just mean not being active. Prolonged sitting has been shown to be a risk factor for poor health, independent of how physically active someone is. Physical activity is still important, but if two people are equally active, then the one who goes home and just sits without getting up is going to be at greater risk of poor health than one who goes home and potters around a bit as well as sitting down. Now, we do know that not all sitting is bad. We all need to rest sometimes. And we sit down to do things that are necessary and important to us. Eating, driving, visiting friends, listening to people talk to you about how bad it is to sit, and so on. And that, as the title of our project says, we are interested in understanding the sitting patterns of older people, why and when people sit, as well as how much. So our problem as researchers is we don't know, there are some things we don't know very much about, and one important thing that we don't know is what sort of health, lifestyle and environment in the past might influence how people sit now, and that's where the Lothian Birth-cohort comes in. Seniors USP data collection will be fitted in alongside the next wave of LBC 36, and we'll be including some questions about sitting in with the rest of the wave questionnaires, but we'll also be asking people to wear an activity monitor for a week, so we can measure how much people actually sit. We'll be using the Active PAL monitor, which is worn on the front of the thigh, and is the orange box on the left-hand side of the slide there. It's fixed to the front of the thigh using sticky pad and goes underneath your clothes. And we'll be asking people to wear it all the time, including overnight, for a week. I've personally worn the monitor quite a lot, and generally I find I forget I'm wearing it. The monitor is going to be covered in a waterproof dressing, so you can bathe, shower, or swim as usual. No, not quite yet. The monitor measures the angle of your thigh. So when you're sitting down or lying down, the monitor knows that your thigh is horizontal. And when you're standing up or walking, the monitor knows you're upright. And what we get out of the monitor is the picture on the right-hand side of that slide. Each of these lines represents an hour of the day, with the minutes spread out along. All the yellow colour represents time spent sitting, the green is time spent standing up, and the red is time spent walking. So you can see that this particular person sat for longer in the morning and was more active in the afternoon, with shorter sitting periods. And from this information, we can work out how much time people spend sitting in total and also the pattern of that sitting. Okay. From this information, if we get a lot of it for a reasonable number of people, we're going to allow us to answer two main questions for the study. The first is we'll use sitting time from the monitor and compare it to the questionnaires, so we can find out how accurate the questionnaires are and also which one's best. And secondly, but most importantly, we'll use the rich and detailed information that's already been collected about the LBC 36 to try and identify what are known as the determinants of sedentary behaviour. That is, what are the common factors of people who sit in a particular pattern, those who don't sit for very much, or those who sit for a longer time. In the future, we would hope to use that information to help design interventions, letting us know what we might be able to change or what it's appropriate to change to help people reduce unhealthy sitting. Okay, that's my 5 minutes. Thank you very much for listening. I'd just like to say I'm personally very excited and also feel very privileged to be able to work with the Lothian Birth-cohort on this study. Thank you very much.
Thanks, Philippa. That's excellent. So again, that will be an optional thing, but we hope you'll be interested in doing it. We've already found that being active is one of the things that does promote better thinking skills over age and even seems to be effective in stopping the brain shrinking and causing these connection damages as well. But actually to have a measure, not just a self-report of how much people are moving around will be fantastic. Now, next thing, I'm going to ask up in a minute Caroline Brett to speak, but before that, anybody born on the 1st or 2nd or 3rd of the month? Anybody born on the 1st, 2nd or 3rd of the month? Keep your hands up for a minute. Anybody born on the 1st of February, April, June, August, October or December? The 1st of February, April, June, August, October or December. Okay, hands down. When we came across these data that form the basis of all this, the Scottish Mental Surveys, it wasn't long before one of the things I personally saw was that there were some folders in the basement all locked away, and I just couldn't believe it, was that for the people born on the first 3 days of the month in Scotland, they had extra sociological information collected when they were children. And for the people born on the 1st of February, the 1st of April, 1st of June, I've started so I'll finish, the 1st of August, 1st of October and the 1st of December, they had 16 years of yearly interview data collected. And it's been a dream of mine. Now you probably think, oh, you took on enough when you did the Lothian birth cohorts. But I've always had a gleam in my eye that I wanted to follow up these cohorts later on. They're called the 6-day sample, people born on the 1st of the even numbered month, and the 36-day sample. And the researcher who's helping me do that and is going to speak about it today, the 6 and the 36-day sample, is Caroline Brett. So Caroline, come and say a bit more about these studies.
Well, I don't really have much to say now that Ian has basically described both of these new samples. So I'm Caroline, and some of you may have met me at the Wellcome Trust when you came to do your clinic visits. Both the LBC 21 and 36, actually, I did both visits. Anyway, so moving on. I've never spoken to this number of people before, so bear with me. So I'm going to talk about these two new really exciting projects, which are, as Ian said, the 6-day and the 36-day sample. So to understand a bit more about who these people are, many of you are in the audience today, we have to go back to 1947. So here we have the whole population of people born in 1936. It's about 75,000 people. And here we have a smaller group of 70,000 people, and that's all the people who took the Murray House test. Now, of course, about 5,000 people were actually off sick, or they were needed at home, or various reasons they didn't actually take the test itself. Now, the 36-day sample, as Ian said, were those people born on the first 3 days of every month. There's about 7,000 people, and they were taken from both groups, of course. And within that, we have, this other group, they were also interested in the twins born in 1936, none of whom I think are in the LBC, but but still it's a whole other group. One pair, apparently, yay. Okay, and then within the 36-day sample, we have the 6-day sample. There are 1,208 people in this group born on the first day of every second month. So just to highlight, that's the group that we are mostly interested in this study. So what was collected on these people? Well, you know the original Scottish Mental Survey was looking at your intelligence score on this Murray House test, but also size and position in family, so whether you were the first of 3 children or an only child or what have you. They were interested in how that related to intelligence. They were also interested in how your intelligence when you were children related to a whole load of different background factors. So this is where the sociological schedule came in. This is the schedule. You can't read it, obviously, but it's 4 pages, and it was filled in mostly by your head teachers, the people in these 3 groups. And this included all this sort of information. So in addition to the family and intelligence, it was birthplace, school attendance, father's job, occupancy rate in the home, mother's age, and some health variables, height, weight, and physical disability. The 6-day sample, on the other hand, many of you are in that sample. You may remember being visited every year by a home visitor who asked you a whole load of questions about your life and how you're getting on. And this is just one person's folder. The folders were actually shown in the film, I think they featured, which was lovely to see. Okay, so all of this additional information was collected. One of the things that's wonderful about the Lothian birth cohort is that you have all this huge information that we've asked you. But of course, sometimes when you ask people about things that happened 50 years ago, it's quite difficult to remember. So it's wonderful to have the actual information from the time about all of these things. So about life events, hobbies and sports, ambition, and of course your family and occupation. So what did we do? Well, we found these 1,208 people. And those who were still alive and living in Scotland and England and Wales, we sent them a wee kit, which some of you may have received. The reason we did this was because they are all over Scotland and quite a lot are in England as well. So we really can't get them all. It's too much to ask to get them all to come to see us in Edinburgh and do all the testing that you all have done. And so we had to do it all at a distance. And so we sent this box. It had a very large questionnaire. It was much bigger than the questionnaire that you guys have all filled in, and it asked a lot of background information, personality, well-being, occupation, etc. And we also gave them some physical testing measures to do at home on lung function. We actually asked them to provide saliva samples for genetic analysis that went to the Wellcome Trust as well, and cortisol, which is a stress hormone. Okay, so that was the 6-day sample. The other thing we did was I telephoned them all, and all the people who agreed, and asked them those questions that John was talking about earlier about, can you tie your own shoelaces, get on a bus, that sort of thing, and did some simple cognitive tests over the telephone. This is my view from when I'm doing the telephone interviews. I have a lovely view of McEwan Hall, which is just over there. We have some sub-studies as well. Certain groups within this sample are going to be interviewed at home about how they think about the ageing process, and their what's called narratives, basically their life stories. We're also going to ask some of them for a sample of their urine, that'll be nice, and others we will ask to come to see us in Edinburgh at the Wellcome Trust and do the same tests that all the LBC have been through, just to check that the information we got from a distance was pretty much correct. Okay. So Ian mentioned earlier the people born on the first 3 days of every month. That's the next stage, which we are in the process of doing at the moment. So a lot of you will have received something from us quite recently. And we are asking the 36-day sample to fill in a shorter questionnaire. And if they're willing, they will do this longer bit of testing with the physical testing and the telephone interview. So that's going quite well at the moment. We've sent out about 2,000 invitations, so I was getting a sack of mail every day for a couple of weeks. So that's our 2 new cohorts, the 6-day sample and the 36-day sample. There you are.
One of the things that, again, you suggested to us and that we have set up is the Lothian Birth-cohort 1936 brain tissue bank. Brain tissue banks that store brain tissue from people who have gone through healthy cognitive aging are rare to almost disappearing. And when very bright young scientist was recruited by the University of Edinburgh, as it is wont to do, Tara Spires-Jones came along. She was immediately attracted by how rare and valuable this was, and so Tara is going to tell you something about that now. So Tara, can I ask you to pop up and say something about the brain tissue bank?
Hi, first I have to say my name's Tara, and it's such an honor to speak with you all, because one of the reasons I moved myself and my kids, my husband, from Harvard Medical School to Edinburgh was because I saw Professor Deary at a meeting in Paris a couple of years ago, and he spoke about you. And it's incredible. What you're doing with each other and with the team is truly incredible. So I'll tell you how what you're doing will fit into the research I'm doing and why I think it's such a valuable resource that you're providing for us. So you know about the brain. The brain's a big organ, takes up most of your head. And Professor Wardlaw and Professor Deary have talked about the grey matter and the white matter. And what I focus on are the tiny little connections. So in the grey matter, the part on the outside, you've got cells called neurons. Those are the brain cells. And those neurons send really long wires, that's the white matter, to other parts of the brain where they make synapses, these tiny connections. So the synapses are the way the neurons talk to each other. And neurons, the brain cells, you've had all the neurons in your brain pretty much since you were born. We get very, very few new neurons in our lifetime. Pretty much we get them when we're babies, and we're stuck with that, and we lose some when we knock our heads, or if we drink a little too much, occasionally we might lose a neuron. And you might think, how are we able to think and adapt and learn new things if the stuff in our brain is static throughout our lifetime? And the key is in these little tiny connections, the synapses. Because synapses, the connections, you can get more of those as you learn. You can lose some, you can change them, and that's how we form new memories and how we recall old memories, is by changing the strength and numbers of these connections. So synapses are really essential for learning and memory, and this has been known from a lot of different kinds of studies, particularly in models, but what's been very difficult is looking at synapses in detail in humans. And this is because they're very, very small. So a single synapse is about 1/1000th of a millimeter in size. These are very tiny, and even with our lovely microscopes, like you can see a picture of my microscope, my third child, on the thing here, even with these wonderful microscopes, we can't really analyze synapses. It was just not possible in the past to do this in human tissue. So what my lab did when I was still at Harvard Medical School was take a new technique that we call array tomography, and this allows us to look in really great detail at the connections in the human brain. Now the problem with it is we can't do it while you're alive, so I get the sad chore of when Ian's phone rings, I get the next call from the pathologist saying we have a case. And it's one of these sort of laughter through tears because I'm so excited about the project, and at the same time it's so depressing. But I want to tell you we take teeny tiny samples of the brain, less than a fingernail size, from different regions, and the pathologist gives them to me, and I take them back to the lab and embed them in plastic resin. And then this plastic, we're able to cut it really thin. I mean, thinner than deli slices. These are 70 nanometers, so that's really on that 1/1000th of a millimeter scale. And this way, we can take pictures of each of these individual sections and build up a nice three-dimensional image of these connections in your brain. Now, why do we want to do this? Why is this macabre woman trying to take pieces of human brain? The thing is, we don't know what causes the synaptic changes. We don't know what causes the decline of memory in some people in aging. And what I've been able to find out in my previous lab, where I worked mostly on dementia, is that these synaptic connections are really driving factors in dementia. But we don't know if the synaptic connections are driving factors in the way that you age, whether you age cognitively well or poorly, whether your thinking skills are good throughout your entire lifetime or not. I think that these synapses may be crucial for that, but we just haven't been able to answer that question in the past. So I wanted to come here because now we have that opportunity. We know, Professor Deary knows, and you know, all this amazing stuff about how your thinking has progressed, how your cognition and medical history, and all of these facts about you from your lifetime. And now we'll be able to compare those with the connections that are left in your brain post-mortem. And we'll be able to answer some of these essential questions about really what makes us human, what makes us able to learn and think and remember. So I have to say a huge thank you, because this is a truly unique opportunity. And I think we have the potential to make a big difference and to find out some really incredible things from you. So thanks a lot.
So we could have had lots of these excellent 5-minute talks, but you see how you're attracting the next generation of really bright scientists to come and work on extending what we're doing in the study. So let's do something different now. Do you know who it was that actually invented the mental test that you all took in either 1932 or 1947? It was a man I have become fascinated, obsessed by, Professor Sir Godfrey Thompson. He lived at 5 Ravelston Dykes. He's 10 minutes walk from where I live. And he was, I'm not going to say too much, Emma, don't worry. He was Professor of Education here, and I will say this one thing, though, is I still have my heart skip a beat when I think I might have missed the telephone call in 2008 when his great niece phoned me and said, "We're clearing the house in Ravelston Dykes. There's some stuff by Godfrey Thompson here. Do you want to come round and collect it?" And I might not have been at my desk that day and not got just about the only half-decent archive that Godfrey Thompson left behind, because he was such a fantastic educator, statistician, and psychologist, and almost now forgotten. So Emma, another fantastic team member, is appointed to Wellcome Trust programme where we're trying to revive the memory of Professor Sir Godfrey Thompson, who is the reason that you're all here today. So Emma, come and tell us about Godfrey Thompson and your part in the project.
Okay, so that's obviously not Godfrey Thompson, that is me. As Ian was telling you, he rescued the records essentially, but he also did a very important part of the archivist's job for them. As archivists, people assume that we keep everything, when in actual fact we have a careful selection process, so we decide what we will keep and what we won't. And in going to Ravelston Dykes and rescuing the material, Ian actually did that job for us. From the mass that was in the house, Ian whittled it down to the key items that told us more about the person that Thompson was. So a bit about me. I've had quite a varied background. I have worked with medical records, which included ultrasound records and nursing records. I've worked with textile records, industrial records, and maps and plans. And I didn't know anything about any of them when I started, and I just kind of accrued the knowledge as I went along. This project has been a bit different in the sense that I've had the help of Ian and Caroline to make sense of what's been in front of me, which has been a godsend. Ian and Caroline have been only too happy to share their extensive knowledge of Thompson, gained both through the LBC studies as well as their extensive historical research. The main thing that this has helped me with is in creating a catalogue of Thompson's collection. A catalogue is much more than an inventory. What we hope that it will do will allow researchers all over the world to get a really good idea of the potential within these materials and what they will find if and when they come to see them. For someone like myself who has a limited knowledge of intelligence testing and psychology, working with Ian and Caroline has allowed me to interpret the records, and it's allowed me to create a catalogue that can be used by researchers from all backgrounds, but yet still preserves and explains the complexity of what Godfrey Thompson was doing. And it also tells us about the complex relationships, both personal and professional, that he held throughout his career. For me, the best part of my job is making new friends. A lot of the friends that I make happen to be long dead, like Thompson, but that doesn't mean that I like them any less. It's a great privilege for me to be able to get to know someone in that way and to see every facet of their personality laid out before me. It's really wonderful. Thompson's technical records are absolutely fascinating, and the collection contains plenty of them. But for me, the parts of the collection that really get me excited are those that tell us a bit about the sort of person Thompson was. The photographs that you can see above are from photograph albums, and we've got a lot of those in the collection. I particularly like the middle one because he looks a bit rakish with the cigarette. Now this is from my absolute favourite item that I've worked with. It's an album that was gifted to Thompson by the Polish students in uniform. The Polish Students in Uniform was an initiative very similar to the Polish School of Medicine, and the Polish School of Medicine aimed to educate Polish soldiers and refugees in Polish, and they could get Polish degrees during the Second World War. The album itself, as you can see from the images, is a beautiful, beautiful object, but for me what makes it so special is that it's a real snapshot of life at Murray House. It stands in quite a contrast to the usual institutional records that you get that are usually very kind of straight-laced group portraits. And you can see that just from the portraits of the lecturers there, it's almost like they've just kind of bombarded them with a camera and they've had some real fun. And I just think that at a time of sadness and uncertainty in the students' lives, it's really wonderful that they took the time to make that. I'll just read you out the inscription in the front. It says, "Please accept this small gift from the Polish teachers in uniform as a token of their deep gratitude and an appreciation of the splendid work you have done in helping us, the many kindnesses, the great patience and understanding in which you have shown throughout will never be forgotten." And I really find that quite moving. If you would like to know more about the work that I do, I'd be thrilled if you would visit my blog. And you can also contact me if you have any questions. I've put my email address up there as well. And you can come to my seminar, Ian's seminar really, and Caroline's seminar. I've put some leaflets out in the front if you would like to see them on the red table. It's been a real privilege to speak to you all today. It's been great for me having the chance to work with Caroline and Ian, and I've heard so much about the Lothian Birth-cohort, so thank you very much for listening to me.
Okay, so thanks very much to Philippa, Caroline, Tara and Emma. And it really is heartwarming to see the enthusiasm for their projects as well. And you know, the thing I like about Thomson is this, and probably my favourite object that we took away from Ravelston Dykes. By the way, they knocked that house down not that long after it and rebuilt it, so it wouldn't have been there. So my favourite object is this big bunch of lectures that are handwritten by him and typed. And time after time after time in his public lectures, he said this: the reason he invented intelligence tests was so that poor children could get the education that their intellect deserved, because on the basis of other things, their social background, that just wouldn't happen. So he thought he was giving poor children a chance in life. He could have made the equivalent today of hundreds of thousands of pounds from all the intelligence tests he wrote. He didn't. He ploughed it back into his research team to make better tests. He's a real, well, you can see I'm a fan. So something now, just finishing, about the future of the Lothian birth cohort of 1936 especially. So this was actually for the benefit of Tom Wright, the Chief Executive Officer of Age UK, who've funded us wonderfully until now. And I was going to say, well, you know, there's always this coming up, and Tom's actually gone back to London. So he's not here for my expensive animation. I'll just show you again. There it is. And what a fantastic opportunity we now have. You've been coming along age 70, 73, 76. So let's finish this 8th decade of life. And I'll not be done with you then, okay, if I'm still around. You never know, God willing. But finishing, can you see also what's going to happen when you get to 79? You'll be the same as where we started with the Lothian birth cohort of 1921. And then we'll be going from age 70 to age 92. So this will be a wonderful opportunity. We'll be starting this later in the year, and it'll be, let's be honest, it'll be the same as you did before. So my only recommendation is try not to rehearse the answers to those mental tests too much between now and then. Now, you've heard already about the Seniors USP project expertly from Philippa Dahl, and again, another one of the young researchers we're pulling in. There's another study too that's been funded that's a much, much, much lighter burden called MOOD, Mobility and Place. And it says there it's a 3-year project. We're working with the Art College and the architects and geographers that builds on evidence that how we experience environments influences our mood and in turn our willingness to be active. and it says, working with the Lothian birth cohorts of people, 70s and 90s, will be the first to consider the influence of local environments in which people have resided from childhood. So here's the thing. Do you know that we sometimes get the team to send you out wee questionnaires between the waves of study? So you get tested, then you come back 3 or 4 years later, but between times we sometimes post you out a questionnaire. Well, the simple thing we'll send you out this time is this. Where have you lived during your life since childhood? Because the geographers have got fancy computerized information these days that can measure the quality of the environments in which you lived and can add that to our evidence to see whether that's affected how well your health and your thinking skills have aged. So, for example, that's Edinburgh's green space in 1949. And the geographers have made up something like, they've not done it properly yet because they're not in touch with all your data, but they've made up something like the Lothian birth cohorts to plot each person and how near they are to some green space. So we can do this over time. Look at this next one. That's 1949, and this is how the green space changed between then and 2009. And actually, they've gone back even further to 1910, and it grows even more. That's quite nice, actually. I must have got it exactly the right size, because that's just basically one thing and then another. But yeah, it's good. It's working well. Okay. So the other thing we value is, of course, your suggestions, because you've led to things that we've done before. And so what we're going to do now is take some questions. Slightly trepidation about this, but Simon, yeah, you're looking like they're going to be difficult, aren't you? Yeah, so if Simon Cox comes up, and John, you come up as well, and there's a couple of microphones there. Do we have to do anything? John, are they just going to work anyway? So they're going to work anyway. He said put them quite close to your mouth when you're speaking, and let's do some questions.
Okay, do you want to take the full bunch there?
Are they too hard?
They're pretty tough.
All right. That's too long. Right.
We'll give you those as a
Okay.
backup.
Right. The good thing is, if you sit down there, I can actually throw questions that I don't fancy to these guys as well. John, one for you. You don't need to do anything, John. Stop fiddling. John, what effects, if any, are there on aging if we take statins? It means I can read a question next.
Okay, so we look at what people say they're taking. So we rely on you to tell us what you're taking when you come. And we only know what you're taking at the waves when you come along. We don't know what you've taken in between. We don't know what you took when you were 24. So we don't know all your life history of medication use. We have looked at statins in particular, so it's good we had this question, in the 1921 group. And there's two things to tell you about it. First of all, people who take statins tend to have a slightly higher IQ at age 11 than the ones who didn't, strangely enough. And that's a bit odd. You'd think, well, take statins for when you've had a stroke or if you've got diabetes, the kind of things that we know are associated with socioeconomic deprivation. But in fact, it's the people who have got a slightly higher IQ who take them. So maybe there's a selection bias to start off with, who goes to the doctor, who gets given a statin. And those who took statins actually did slightly better over the years that they, so if we compared their Murray-Hales test, the people in 1921, when they were in their 80s compared to, when you're age 79 compared to age 11, they're actually doing slightly better. Now, who knows whether that's due to the statins or not. But we looked at other drugs, and other drugs didn't have any particular effect apart from drugs that people take for mental health problems, in particular things to calm you down. They weren't so good for you. So most drugs have very, very little effect on cognitive function, but statins, if they have any effect over the piece, and this is an average effect, so you can't say for some people they might be better, some people might be worse, but on average, they're doing a good thing. So,
thanks, John. Okay, here we go then. John, a couple of medical ones I'm going to answer briefly, and then you can chip in if you want. So it says here, would it be useful for us to get the results of our brain scans? Well, in a way you do, because Professor Joanna Wardlow, who's a clinical neuroradiologist as well, reads studies carefully every brain scan, and anything unusual there gets reported back to your general practitioner, which is part of the consent that you sign up to. So in that way, it is a health check. And we recently, under Joanna Wardlaw's leadership, wrote a research paper that was published in one of the British medical journals, and it was about the incidental findings that we found, because you're an unusually large number with brain scans, 700 or so, and so inevitably Professor Wardlaw saw things, and these were all noted very carefully, and John Starr writes the medical letters that go back to the general practitioners, and how many medical interventions had to be made on the basis of those 700 scans? Precisely 0. Medical intervention, operation, yeah. But there were many things discovered and fed back and treated, but there were no operations based on the brain scans. So, what information is fed back to the GPs?
Hello, that's better. Okay, so on the brain scans, it's quite. I look at all the brain scans, and most of them say normal scan for somebody's age, and it would be absolutely useless to anybody to know that information. The GPs can, some of them, if they choose to, can look on the system and look at the result if they wish to. If you come into hospital, those scans are available to anybody at the hospital. So if you come in and there's a question of had you had something new like a stroke, etc., they can compare your new scan when you come into hospital with the one that we've done already, and that's very helpful. I write to GPs if the blood test results come back abnormal. If they come back very abnormal, which is, I think, very unusual, but occasionally, then I will ring up the GP and they will either get in touch with you or I'll get in touch with you. If I ring you up or if the GP rings you up, it means that something has to be done fairly quickly. That doesn't happen very often, but we do pick up things. And that's a good thing, isn't it? Because you could be sitting there with something you didn't know, and it's good we can do something about it just by chance. Most of the things I pick up are things like vitamin deficiencies or something like that. They don't need urgent attention. They're not going to get into big trouble. It's sensible to get them sorted out. I write quite a lot of letters. I imagine I write letters on, I don't know what proportion of people, but probably between 5 to 10% of people, I will write a letter to the GP. But you very rarely needs anything urgent doing about. Yeah.
Okay, I'm going to do one on genetics and then a humorous one. So what is a genome, please? Well, a genome. Well, each of us has 22 pairs of chromosomes, and then if we're men, we have an X and a Y, and if we're women, we have 2X. And on those chromosomes, there are approximately just under 20,000 genes. And genes make proteins, and that's what they do. And genes come in different flavors. And what we're testing is the differences between people in the genes that have been given to them by their parents, their mother and their father. And we have tested each of you already on your genome, on your genes, in this number of places, 610,000. We ran 610,000 genetic tests on each of you to find how you differ from the rest of us on genetic information. Right, this one says, that's not the one I intended. You referred to smoking, how about alcohol? Well, this is the work of Janie Corley in the team, and I always use this one when I'm speaking at lots of clubs like Third Age and Inner Wheel and etc. and Probus, not just the women. And it's this, that our initial finding in the Lothian Birth-cohort of 1936, now you have to listen carefully here. Now, we haven't got terribly heavy drinkers in the group, okay? But we were finding that the people who were drinking more alcohol in the Lothian Birth-cohort 1936 had better mental skills across the board. I'm afraid you're all getting ahead of yourselves because I hadn't finished. We then looked, remember, we have the mental ability scores from age 11. So just let me rehearse that. So higher scores at age 70 went with drinking slightly more alcohol until we took into account how clever people were at age 11. Then what did we find? We found that relationship completely disappeared. And here's what the story that Janie told actually was. It was that the brighter children at age 11 are the ones who drink a bit more at age 70. And that drinking more has got nothing to do whatsoever with boosting your thinking skills. That's it. Right, John, I could do this one, but I'm going to give it to you. Any exercises to keep the brain ticking over? Crosswords, bridge, Sudoku, etc.?
I think you should do this one, Ian, but I'll just say that you will be, if you are going to practice those names of those people or whatever, that story, you probably do quite well. I have people come to see me in my kind of memory clinic, and they deliberately practice various these tests, spelling particular words backwards, for example, and things like that. Maybe that's just part of their good mental abilities to remember to do it. The bottom line is you can practice specific things and do quite well in those specific things, but it's actually very difficult to do an exercise that improves your mental abilities across the piece. We actually think that actually being more physically active is probably more helpful to you than sitting. So we're sitting down doing a crossword, you're probably better to go and have a walk for 20 minutes instead of sitting down doing a crossword.
Okay. It was mentioned that study of a second language help the brain. Remember, that's under wraps yet. We can't actually officially report that. What about the study of other subjects under long-term learning? Can this help as well? Well, again, one of our younger researchers, Stuart Ritchie, has recently written a paper based on information from the Lothian birth cohorts and has found that on top of how you were scoring at age 11, more education gives you a slightly better edge in your thinking skills in older age. So educate, but it's helping for certain types of skill. It helps those sorts of skills that are to do with knowing more knowledge, words, vocabulary, that kind of thing, and not so much on how fast you're thinking. So it's not a straightforward story. Right, Simon, since you're the study coordinator, and he's awake, good. How selective are the cohorts? Have you any ideas as to why some people choose not to participate and why some people drop out?
It's a very good question. I'll try and keep a potentially long answer fairly brief. They are fairly selective in that, obviously, when we contacted you at the very beginning of the Lothian birth cohort studies, it wasn't compulsory for you to come in. And it seems that actually the individuals of you that did come in and want to take part in the Lothian birth cohort were slightly brighter, even at age 11, than the individuals who didn't. However, over the course of the 3 waves of testing that we've done so far, we often hear people asking, how many of us are left, how many of us have popped our clogs, or actually surprisingly few. In fact, to the extent that Ian sometimes expects far more of you to be popping your clogs than you actually are, so you're certainly defying his expectations. But
that's the last question he gets.
Where was I? You've distracted me.
Sorry.
But actually, as you will have seen from Ian's talk a bit earlier on in the day, it's amazing how many of you want to come back and how many of you do return. Our retention rates are very high. And actually, the majority of people who don't come back are those who either don't fancy it for whatever reason, which is perfectly fine, or those who maybe are a little bit, they're getting ill, or they're not able to come in because they're having an operation or something like that. So that's the general picture as we see it from the coordination side.
Thanks. Will LBC 2136 press for government to have a survey of 11-year-olds in the future? In a way, you've no idea how much I would like the government to be able to test an entire year of birth in Scotland on exactly the same test as you got. But there are 2 reasons why this will never happen. One is some of the questions are rather archaic for today's children. And the second one is it was conducted at a time when you didn't need to ask parents permission to do things to their children. And they pretty much were able to test the entire country because of that. And we certainly had only one participant I can remember who said their mother actually did storm up and complain about the thing, but these days you wouldn't be able to do it. However, there are other cohorts in the British Isles that are younger than you and that don't have necessarily the same or as detailed information. So there is a cohort born in 1946 that learned a lot from the 1936 born people here. There's a cohort born in 1958. There's one born in 1970. There's a millennium-born cohort, and they're about to start the new life cohort, people born in 2013. So there are ones coming up behind you, but none which have got into the age range that's of interest to us in terms of over 65, but they will be coming through, not doing exactly the same, but some of them do have early life information as well. But there will never be another, I don't think there'll be ever another entire year of birth testing. I mean, I think you'll be able to say, you are unique, because I don't know as yet anywhere in the world that's ever done this before. And these other cohorts I've mentioned typically are things like week of birth cohorts rather than entire year of birth. I'm just trying to get the time because it's in the thing. Right. Oh yes, right. Another one for the boys on the bench there. John, is there evidence that being part of the study is keeping people well because of the interest and attention of being in the study?
Well, I don't know. It's a very simple answer to that. I mean, there is evidence in general that people in cohorts do quite well. People in trials do very well, because it's difficult to say in a cohort study because we don't know what would have happened otherwise. But when you look at people who take part in trials, that's when one group get a specific treatment and another group, say, get placebo. People on the placebo side seem to do better than you would expect just because they get a lot of attention. It's actually quite good to be part of a study. And coming back to my previous answer on health, if you come up to the study and you have your blood test done or whatever it is, and you get detected you've got a vitamin deficiency or something, that wouldn't have happened had you not come. You know, it would have happened maybe a bit later. You might have presented with anemia or some vitamin deficiency. The fact you turn up means you actually get screened for a whole range of things, and therefore you probably do stay a bit healthier than otherwise. And that's part of a dilemma of following up a cohort, because in a sense we don't really want to interfere with you, but we have an ethical duty to care for you, so that if something does turn up that is likely to be detrimental to your health, we will inform your GP and you will get that sorted out. So in a sense, you do stay healthier than you would otherwise, partly because attention is paid to you.
Right, a couple of quick ones then. Any possibility of individual profiles? Well, my answer to that is, well, we do that already insofar as John's a medical practitioner, and any results that come back from the laboratory or from Professor Wardlaw that are abnormal do go back to the general practitioner. And secondly, and I'm going to apologise to my team here for saying this, but those of you who have asked for your childhood scores, I have taken the attitude from the beginning of these surveys that these are your data and not ours. And so I think you get them if you ask for those childhood ability scores on the Murray House test as well. But insofar as individual profiles for all the other data, it just wouldn't mean anything. These data are interesting. All those mental tests I showed you, they're interesting with regard to how everybody's doing with respect to each other, but given in isolation, they wouldn't mean anything. So once again, if something's important medically, it will be sent to your general practitioner. That is, for me, an individual profile. Simon, here's one for you. Are you not just lucky or unlucky with the brain you're born with? That's a real googly, actually, isn't it? Yeah, you might want to take an hour or two to think about that. So well, are you just lucky with the brain you've got? Yeah.
I think
not you particularly.
It's a great question insofar as it highlights the strengths of the studies that we're talking about today, the studies you guys are taking part in. We have a pretty good measure of how well your brains are doing at age 11 years old. And we're now... Oh, sorry, John, are you going to go and... Thanks very much. And we're also now identifying what kind of things over 3-year periods from your 70s, what kind of things are changing in your brains now. And we can look at those changes with respect to how good, essentially, your brain was when you were 11 years old. That's an incredibly rare thing to be able to do. Take into account as well that we can look at your genes, which is really not just about how good your brain is, but how good is the body that you're born with, what's the nature of the building blocks on which your body is based. And we can examine that with respect to change of your brain and its functioning and your thinking skills. So a really, really good question. The short answer, to an extent, yes, it is. But it's certainly not just down to the brain that you're born with. There are things that we're able to identify that may be able to ameliorate possible changes as you age.
Okay, and I want to say something which I think is very important. You heard Ian talk about differences. Actually, it's remarkable how very similar you are. The actual abilities are actually very similar if you consider how complex the brain is and the fact that you're so similar now in your 70s to how you were at age 11. The actual amount of, the proportion of variability in your mental ability scores in your 70s explained by your age 11 mental ability is considerable. It's nearly half. So that I see people in my practice who have severe learning disabilities, and their brains, you would say they are unlucky who they're born. But you here haven't that. You actually fall within quite a narrow range. So although we've talked about differences here today, I think it's important to emphasize that actually you're all doing really, really well. And whatever the differences are, they're not that big. We're looking at small differences that will give us clues as to how people change as they grow older. But you could have been born and could have been very unlucky and probably wouldn't have survived to the age you are if you'd have had a very bad brain to start off with.
Okay, so quick ones. Deafness, somebody asked about, we are looking at hearing and its contribution to broader health and cognition in the 21 and 36, but we haven't written up anything on that yet. Balance, similarly, John, we've got information on balance, but we haven't written that up yet. Interesting question here about what's the contribution of extracurricular and committee work in terms of health. Funnily enough, that is something we're looking at, but in the 6-day sample study, one of the professors that's helping us on that is particularly interested in community contributions and how that contributes to ageing, so that's not out yet. Any exercises that keep the brain ticking over, crosswords, bridge, Sudoku? Well, you'll remember Alan Gow very well, and Alan, that was one of his interests, and broadly speaking, what he found was that generally speaking, the people who are doing better on thinking skills are doing more of these things, these intellectual activities, but that once again is explained by IQ score at age 11. It's the bright youngsters that are still doing intellectual things in old age, and they don't seem to be in our study, boosting brain power in older age. And just lastly, what about the body and mind link? One must affect the other. Absolutely right. And just to echo what John said as well, one of the genuine surprises we have got from studying the material, having this very rare thing, this mental ability test from age 11, is how much that contribution, and that's an aspect of the mind, contributes to health and even survival in older age, and also other things like personality and how they contribute to well-being and health in older age too. And that's about all the questions. So I'm going to wrap up a couple of minutes early to say, obviously, thank you very much for coming along today, to thank some of our funders and areas that we work in, and to invite you all back soon, once we've seen you again, to another wonderful celebration like this, and to thank the speakers today, John Wardlow, John Starr, Philippa, Tara, Caroline and Emma as well, and all you for coming, and Simon and the team, what a fantastic job you've done and not made me worry at all about the arrangements by just getting on with it. So for you and the team, sincere thanks. And John, once again, John Williams at the hall here and his staff always works very well. And we're finishing by not just having a film, so you have now produced a film in the Lothian Birth-cohort. There's now also a book called Lifetimes, which celebrates something beyond the science. We collect numbers, that's what I'm interested in. Your numbers of how well your brain's doing, your health's doing, your cognition's doing. But Anne Lingard, gifted writer, came in and wrote the stories, the individual stories, the personal stories of some of the participants in 1921 and 1936, and some of the scientists. This is unusual for us. You don't usually get the focus turned on you, except by the artists and the filmmakers and the writers that have come in. So, Robin, the arrangements are... That's very simple. Good, that's the way I can cope with it. You pick up the Lifetimes book as our gift to you to celebrate the Lothian Birth-cohort, 1921 and 1936. And you'll all be on the telly, STV, 6 o'clock tonight, so you better get on that bus. Thank you. Cheers.